So, hi class, we are going to um, learn about chapter 5, and this chapter 5 or chapter 5th is about recombinant DNA technology. So the question arises that why it is called as recombinant DNA. The reason it is called as recombinant DNA is because we fuse two DNAs into one. And why do we do this? We are doing this for, number one, to increase the productivity. And when you increase the productivity, there are high chances that you reduce the cost. And third, you cater to the society globally. Now, thing is that why would you fuse to DNA? We would fuse to treat human diseases, human diseases. And many diseases are now having a molecular biology based approach. Why? Because it can be done. It can be done easily in the lab settings. Can be done in the lab setting. So to revise, recombinant DNA means fuse two DNAs into one. And why do we do this? To treat human diseases. And this is a molecular biology approach. And this can be done under lab settings. Now, what are these two DNAs could be? Again, coming back to the same question. So these two DNA could be a human insulin gene and bacterial plasmid. Now, human insulin gene, why? Because we know that diabetes is highly prevalent now, and is now starting from a very young age. And of course, it was a disease of elderly people, but now because of various reasons, diabetes is happening in young kids all over the world. So the only way it could be treated is by insulin injection, one of the form of diabetes. And so insulin has to be not only available in US, but every corner of the world. And so we have to not only produce for US, but for global demand. So now, why recombinant DNA technology? So if we harvest human insulin from humans, let's say human cadaver or human beings, the amount will be very, very small. We don't produce too much insulin. That's the magic of our body. When there is a sugar up and down, our body produces insulin, but it is in a very tiny amount, and that cannot be commercialized. And we cannot do research on human body because that's unethical. So we need an alternative strategy, and that alternative strategy is recombinant DNA technology. And so we fuse two DNAs. Of course, since we are producing insulin, so you need human insulin gene from human chromosome, and then you need bacterial plasmid. Now the question comes, why bacterial plasmid? Okay, so why bacterial plasmid? So what is plasmid? So let's say this is a bacterial body, and you know bacteria has its own DNA that is called as nucleoid, right? Bacteria don't have nucleus, but this is the main DNA. Bacteria also has additional circular DNA. This circular DNA, this additional extracircular DNA is called as plasmid. Yes, plasmid. Now, what are plasmid? As the shape suggests, it is circular. Second, it is like any other DNA. It is double stranded. But in addition to being circular and double-stranded, plasmids have two more features. Number one, it has restriction enzyme site. I'll come to that later. And fourth, it has antibiotic resistant marker. Okay? So these are the four essential features of plasmid. Now let's go a little bit more deeper. Now, so this DNA, this is the major DNA, right? And the plasmid is a extra chromosomal DNA. So this is the main DNA. This is the main DNA. Main DNA means if this is lost, bacteria will die. The plasmid DNA is called as extra chromosomal DNA. So this extra chromosomal plasmid DNA, if it is absent, bacteria will not die. I repeat, main DNA, if it is lost, bacteria will die. If extra chromosomal DNA plasmid is lost, bacteria will not die. It is not important. Now, one thing which is very important that is that both when the bacteria divides, when the bacteria multiplies, both the main DNA as well as plasmid DNA replicates, means DNA duplicates, but they do it independently. So another important feature is that plasmid DNA, they have their own origin of replication. Means both these DNAs, main DNA and the bacterial plasmid DNA, they both replicate independent of each other. So to review, it is circular as the shape suggests, it is double-stranded, it has some unique sites which is called as restriction enzyme site. That's very important that you should remember. And the fourth thing is that it has antibiotic markers, right? Also, I would like you to recall that whenever, let's say, for example, we have throat infection and it comes positive for any bacteria, streptococcus, then, we go to health clinic and urgent care or physician, they if we are carrying bacteria in our throat after testing, then the physician prescribes antibiotics. When we take antibiotics, bacteria dies, but bacteria doesn't die all of a sudden. We have to take the course of antibiotic. We have to finish the course. That the physician always reminds us to finish the course. If we don't finish the course, then it can give rise to antibiotic resistant bacteria, which could be more aggressive to cause disease. So bacteria are very sensitive to antibiotics, right? So think about it. How bacteria becomes resistant. So one of the mechanisms that helps bacteria to become resistant to antibiotic is these plasmids. So these plasmids, they carry antibiotic resistance gene. So if somebody gets infected with bacteria, which has a resistant gene, which has a plasmid, which carries resistance to, let's say, amoxicillin or to augmentin or all these antibiotics, then that person will be not treated with that antibiotic. So physician has to do antibiotic sensitivity test. So maybe that patient will be given double antibiotics or multiple antibiotics. So antibiotic resistance is important thing. But most important thing of this chapter is that here scientists are using or scientists have developed bacteria as a lab agent, as a lab tool to produce therapeutic molecules such as vaccines, such as insulin. So bacteria, which is dangerous, which causes disease, scientists have tamed the bacteria, have converted the foe into a friend, such that we can utilize bacterial plasmid, bacterial enzymes to develop therapeutic and diagnostic tools. So we will take an example of human insulin gene. Okay, human insulin gene, it is important for diabetes, right? So people who don't have the insulin in their bodies and sugar is not regulated, they tend to develop diabetes, which could be dangerous. It can cause many things. So we need to inject or we need to add insulin to their body through various ways. So let's take an example of insulin. So for sake of understanding, let's say this is a human chromosome. So for sake of understanding, so let's say this is a human chromosome, and within this chromosome, this particular area is within this chromosome, this DNA is this DNA or this gene makes insulin. So what scientists can do, they can cut these genes, cut this gene from here, and they can cut this gene from here, and they can take out the insulin gene. So let's say they take out the insulin gene from this portion, right? They took out the gene, and then they will put this gene by technique into a bacterial plasmid. So what happened? Human gene, through techniques, through technology, has now been added into the bacterial plasmid. Okay? So now this bacterial plasmid has the human gene. Then what scientists will do, they will transport this plasmid containing human gene into the bacterial body. Right? So this bacterial body will now have a plasmid, and this plasmid will contain human insulin gene. This plasmid will contain human insulin gene. Now we know that the bacteria, they divide very fast. They divide very fast. Within hours, bacteria can depend, if you provide bacteria nutrition, within hours, they can fill a room and, environment. So this bacteria containing human insulin is put to divide. And when the bacteria divides, it is not only producing bacterial protein, but it is also producing human insulin. Human insulin. So that's the briefly recombinant DNA technology, to cut this human insulin gene and then add it to the plasmid. And then this plasmid is taken back into bacteria, and bacteria divides and divides and divides. And then while bacteria is dividing, it is producing human insulin. This human insulin can then be scaled up at an industrial scale, and then it can be packaged and can be given to pharmacies or clinics. Now I used some words. What words I used? I used one of the word that I used was to cut the human insulin gene. And then I used that, the second word is that to put or to paste human insulin gene into bacterial plasmid. So I use two words. First is cut. First is cut, and the second was paste. Two words. So how this is possible to cut and paste? So there is another protein that bacteria produces inside its body. That protein is called as restriction enzymes. This restriction enzyme can cut the DNA. So restriction enzyme is just, in other words, it is called as molecular scissor. Restriction enzymes, they are called as molecular scissors that can cut the DNA, but they cut the DNA at a very specific site. Right? So I'll come to that, what a specific site is. And then how it is pasted. The DNA, the human insulin gene, is pasted by help of another enzyme, which is called as DNA ligase. So I repeat, cut the human insulin gene using restriction enzymes, and human insulin gene is pasted into the plasmid. This plasmid is inserted into bacterial cell, and as bacterial cells divide, human insulin is produced that can be packaged and can be delivered to clinic or pharmacy. So this is the basic, this is doable. There are many companies which are thriving on producing not only insulin, but flu vaccines, many other vaccines, many other growth hormones through this strategy. So this, what happens now, where was the fusion DNA? This was a fusion DNA, right? Human insulin gene and the bacterial gene. So just like you have fusion music, you have old songs with new music, or vice versa, right? So two different DNA fused together to scale up the production for what? To treat disease. So that's the technology, right? So let's go back to the slides. So recombinant DNA technology. To be able to explain what cloning of a gene is. So to go back to the whiteboard, so what is happening? We created this plasmid, right? We created this fusion plasmid, right? And this plasmid was inserted into this bacteria. Now this bacteria, from one bacteria can be converted into million of bacteria. So when one bacteria generates millions of bacteria which are identical, that is called as cloning. Cloning means one object can create millions, trillions of identical objects, right? For example, a photocopy machine. You can take one page and you can just press the machine, Xerox copier, million times. Each copy will be very identical. So that's an analogy for cloning. So this bacteria plasmid, which has human insulin gene, when the bacteria is multiplying, that is also multiplying from one copy to million trillion copies. And that is called as cloning. So we be able to, what's cDNA will come later, be able to explain what a plasmid is and why it is used, that I just explained. To be given an example of how recombinant is useful, I gave an example of a human insulin gene, and then we will cover in this part. Okay. So what is a plasmid? As I mentioned to you, plasmid is an extracellular DNA, extracellular small DNA. This is the major DNA, main DNA, which has 1,000 to 5,000 genes. But plasmids, they are small, circular, double-stranded, and these, they have origin of replication. So what is an origin of replication? Origin of replication is the point within the plasmid that generates duplicate plasmid. So that's the point of replication. I don't want to go into details. So origin of replication is the site at which plasmid starts the process of duplication. So one plasmid can give rise to two daughter plasmids. The second term I discussed was antibiotic resistant. So if a plasmid has antibiotic resistant, this bacteria containing this plasmid can survive in antibiotics. So let's say if I get infected with streptococcus, which has plasmid that carries resistance to amoxicillin, then if the physician prescribes me amoxicillin, it will not work because I have an antibiotic resistant bacteria. And where this antibiotic resistant gene present, it is present on the plasmid. So the physician has to change the strategy, and it happens a lot. That's why use of antibiotic is not very popular, despite, it's very important tool for medication. So and then coming to restriction enzyme. So restriction enzyme is the enzyme that cleaves or cuts the DNA at a specific sequence. So here in this example, we are taking an example of an enzyme called as EcoR1. And this EcoR1 is produced by a bacteria called as E. coli. And this EcoR1 enzyme can cut this DNA at a specific sequence. What is this sequence? GAA-TTC. So look here, it's very interesting. What is interesting here? Look at this sequence. If you look at this sequence, you will figure out that the DNA in both the direction, it reads the same way. What do I mean? So if you read from this side, GAATTC, and you read from this side, so what do I mean by this side? If you read from this side, it reads GAATTC. And then again, if you read from this side, it reads the same. So they are symmetrical. So this is a very special DNA piece of sequence. They read same. So just like the word, such as, words like madam, right? Words like madam, they read same from both the ends. So M-A-D-A-M, M-A-D-A-M. So from both the ends, it reads the same. Such words are called as palindromes. And in terms of DNA, it is called as palindromic sequence. So these restriction sites are palindromic sequence. They read the symmetrical from both the end, going from A to Z, going from A to Z. And these restriction enzyme can cut the DNA at a very specific site. When the restriction enzyme cut the DNA, these cut site can, on the plasmid, can serve to insert the target gene. So target gene in our case was insulin gene. Insulin gene. So insulin gene, and this is the plasmid. So the target gene can be inserted into the plasmid through cutting and pasting, right? So this is the same way. The plasmid is cut, and then this plasmid could be inserted into the, this target gene could be inserted into the plasmid for bacteria to divide. And then the bacteria with the foreign gene is called as recombinant DNA. The bacteria with, or a plasmid with foreign gene, insulin gene, is called as recombinant DNA technology, recombinant DNA. And then this could be scaled up by selecting those bacteria which has antibiotic resistance gene. So the concept is that let's say you take the bacteria, right? You take the bacteria which has only the main DNA, right? There is no plasmid DNA. Now, if you take this bacteria and put it in a petri dish, put it on a petri dish. Let's draw a petri dish. So if you put it on a petri dish and you put it on a petri dish, so, and this petri dish has an antibiotic called as ampicillin, right? So this bacteria will not grow. So this bacteria will not grow in ampicillin, and this bacteria will die. So this bacteria will die because this cannot grow in the antibiotic. This doesn't have plasmid which would resist ampicillin antibiotic. On the other hand, if you take a bacteria, if we take a bacteria, right, that in addition to main DNA also has a plasmid circular DNA which carries an antibiotic resistance, then this bacteria can grow in this because they are carrying antibiotic resistance gene. So human insulin gene containing bacteria, plasmid, will grow in this. This is what is happening in this figure. So that essentially is the chapter on recombinant DNA technology. All right. So with this, we finish chapter on recombinant DNA technology.